In this video, we will discuss about some of the mechanism of the immunological synapses. That means how the immune cells interact with the tumor cells and also with the antigen presenting cells. So we will start with this helper T cells interactions with the antigen presenting cells. In this case, objective is to provide antigen informations from the antigen presenting cells to the helper T cells. So the T cells can develop antibodies against those specific antigens that are presented by the antigen presenting cells. So in this case, this MHC2 interactions with this T cell receptors is important, and this interactions alone is not enough. It needs other interactions also, such as ICAM1 interactions also needs to be there, and also this LFA1 interactions that will and that will and all this all together will cause the activations of this LCK, and that will lead to the polymerizations, actin polymerizations, and thereby trigger the antigen thereby trigger the activations of these T cells to form antigen specific to form the antibodies specific to the antigen information provided by the APC. And in terms of, and these are the mechanisms in the top rows is the canonical mechanisms that are usually found in the body of the human beings. So in this T cells, CTL, that is cytotoxic T cells lymphocyte, or we can say the antibodies generated after this T cells receive the antigen information from the antigen presenting cells. So in this case, what will happen is that the conformations is done by the receptors, that is T cell receptors, that those antigens are present in the tumor cells or not, and thereby it will lead to the release of lytic granules, and that will lead to the death of the tumor cells. In the case of NK cells, the similar thing happens, but there is one more enhancement occurs, that is uh, lytic granules are also released through the interaction of A and K ligands that are present on the tumor cells when it interacts with the A and K receptors, and also the CD16 that contains the antibodies against the cell surface antigens that are present on the tumors. So this both of this will release the granules, uh, or lytic granules, that will cause the death of the tumor cells. In terms of the CAR T cells, what will happen is the CAR T cells does not depends on too many interactions as the helper T cells. However, it will depends on the antigen information that are expressed on the CAR T cells, and when it interacts with the tumor cells, it will lead to the granule formations, because intercellular signaling pathways are be designed in such a way that it will lead to the lytic granule formation that will kill the tumor cells. And in the CAR NK cells, what will happens? The similar thing happens, but there is like enhancement in the intercellular part that will cause the release of more granules, and this happens because of the ZAP70 activations, and that will lead to the more granules for more granules formations, or more granules released against the tumor cells. And the advanced CAR T cell versions, or the latest generation CAR T cell versions, that is CAR, one of the example of it is CAR PDZ NK cells. In this case, the intercellular signaling has been further enhanced, so such that it can leads to this H script formations, and along with that ZAP70 is also polymerized or activated. So this in together will release more granules, or we can say the intensity of granules will be more compared to the previous two cases against the tumor cells. So these are some of the some of the mechanisms. The top row is about the canonical mechanisms, and the bottom row is about the CAR mechanisms that have been designed by the researchers to target the tumor cells specifically. I hope you understood to some extent how this immunological synapses works. If you need to understand more about their mechanisms in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.